Okay, good morning everyone. My name is Tania Ivorra. I'm from Spain, but I'm working here since 2020. And currently I'm a senior lecturer at the Faculty of Medicine in here at the University of Malaya. Today I will present the part of my PhD student work, okay, entitled Exploring the Use of Black Soldier Fly Larvae in Organic Waste Reduction at Landfills in Selangor, Malaysia. So, for introduction, let me talk a little bit about landfilling, because landfilling is the primary waste treatment in developing countries. And we know that with the increment of the global population, this leads to the increment of the waste generation. So here in some countries, the landfills are mostly unsanitary, and most of the waste that is dumped is composed of food and green waste. So we can apply the insects to recycle this organic waste, and this will help us not only to reduce the waste, but also to get a new sustainable source for nutrition for livestock. Okay, I guess all of you know the start of this presentation and most of the presentation in this forum, Black Soldier Fly, but allow me to talk a little bit about that. It's believed that its origin is in Central or Tropical America. The adults don't feed, however, the larva feed on different type of waste. Food waste, palm oil waste, fish waste, manure or sludge. We can extract from BSF larvae different types of proteins, lipids, and chitin, and also can be applied as an animal feed because they have a high nutritional value. So the objective of this study was to evaluate the ability of black soldier fly larvae to consume various landfill wastes and their conversion efficiency for future applications. For the methodology, we divided in two parts, laboratory experiments and fieldwork experiments. For the laboratory experiment, we use 2.5 kilograms of substrate, and we use different type of waste. The first thing is that we use a control that it was a commercial chicken feed, then food waste that was organic waste, then sludge, and then aged landfill waste that it was a waste that it was already decomposing for 15 years. So all of this coming from the landfill. But also we mix sludge and aged landfill waste in different proportion, but always in a total of 2.5 kilos. The proportion that we used was 25% sludge, 75% aged landfill waste, 50%, and finally 25% aged landfill waste and 75% sludge. And then in total we have 7 different treatments where we apply 1 day old black soldier fly during 20 days, and we observe during 1 month in a grow chamber at 30 degrees. For the fieldwork experiment, we built this type of container. Everything was manual. Our grant didn't have too much money, so we couldn't do it auto. And in this part, what we do is choose the most effective substrate based on our laboratory experiment results, and use 500 kilograms per replicate for this substrate. But first of all, this substrate, we process this substrate with a grinder, and then we place 10 kilograms of this waste per tray, plus 55,000 of larvae per tray, giving a total of 500 kilograms in total per replicate, as you can see here in the image with the trace. So these are our calculations, the one that we did. We focus on the waste reduction, bioconversion efficiency, waste conversion efficiency, and survival rate, and these are all the formulas that we use. So for the results, let's talk a little bit about the laboratory results first. Here you can see the graph with the waste reduction for only six treatments, because the sludge, nothing survives with the sludge because of the high moisture of the sample. So here we can focus on the food waste was higher. The waste reduction of the food waste was higher than the control. However, was less than the previous literature, because in our sample the pH was acidic, but also because of the decomposition associated with microbial activity. On the other side, we have the landfill waste and also the mixture with the sludge that are usually a little bit less than the control. And this is because in this type of waste, we have woody materials, the casting compounds, and harmful materials that could hamper the larval consumption. Here we focus in the waste conversion efficiency and also bioconversion efficiency, and as you can see, both of them are very low. Even in the food waste was lower than the control, and this is because we had in our samples a very low survival rate that impact the final yield of the BSF biomass. And then, as you can see here, the landfill waste and the mixture with the sludge, it was almost inexistent. Because this type of waste are less palatable and digestible for BSF larvae. Okay, and finally, let's focus on the larval survival that you can see in purple color. As you can see, it's very, very low survival rate. And we think even less than other published articles, and we think this because in this case, in this study, we apply a newborn larvae, maximum 1 day old. So as you know, it's a very sensitive stage of the life cycle. So we think that's why we have a very high mortality. Okay, let's go to the fieldwork experiment. As I mentioned before, we choose the most effective substrate that based on all laboratory experiments. In this case, was the food waste that is organic waste, and these are the table. Okay, the table with all results for the 3 replicates and all the parameters that we studied. And here, the last graph is the comparison between the laboratory experiment and also the fieldwork experiment for all the parameters that we studied. In red color, you have the fieldwork experiment, and then as you can see, in all the parameters are higher than the lab work. However, it's especially higher in bioconversion efficiency, in waste conversion efficiency, and also survival rate, showing statistical differences in 3 of them, and even having a survival rate for the fieldwork of 70%, and the lab work of only 10%. Well, to conclude my presentation, I would like to highlight that BSF larvae demonstrated a potential for the valorization of the landfill waste at large scale, but because the waste was transformed into BSF biomass that can be used for different applications. However, as we know, further research is needed to enhance the biomass efficiency, conversion efficiency in landfill waste at large scale. These are the acknowledgements for all the institutions that funded or collaborated in this project. And that's all from my side. Thank you so much for your attention.